Okay. So let's go over the next thing, and that is glycogen synthesis and glycogenolysis. So now, in order to make that, we must have something in the form of glucose 6-phosphate. So imagine glucose is trapped within the cell. Look, all these processes can occur if you trap glucose first. So let's say you have glucose 6-phosphate. First, it must be converted to glucose 1-phosphate. Okay, and this is reversible step. Isomerase will do that. And then glucose 1-phosphate is combined with UDP glucose, and it's converted to UDP glucose 1-phosphate. Okay, and then this is the one that you can add one sugar at a time and to make glycogen. Okay, glycogen then can be broken down. Very straightforward, simple, straightforward. Can be broken down to glucose 6-phosphate. Okay. Sorry, to glucose 1-phosphate. Why am I saying that? Glycogen can be converted directly to glucose 1-phosphate, but sometime in the middle you will have these branches. Okay, branches known as dextrin. Okay. And the branches can also be converted to glucose 1-phosphate, but separately. Okay, so let's just look at it this way. Let's start with this. The enzyme that takes glucose 1-phosphate and converts to glucose 1-phosphate UDP is known as glucose 1-phosphate UDP transferase. Okay. And then the enzyme here, there's two enzymes. One is known as, the major one is glycogen synthase. Glycogen synthase. And the second enzyme here is branching enzyme. Branching enzyme. So total of 3 enzymes in this process, right? Okay. The enzyme on this side that breaks down glycogen to glucose straight. Okay, so we'll break one glucose at a time. It's known as glycogen phosphorylase. Okay, but now there is also a second enzyme that will convert branches into glucose. That's known as debranching enzyme. Okay, debranching enzyme. So debranching enzyme. I'll just put DE, DBE, debranching enzyme. Okay. Finally, we can take glucose and glucose 6-phosphate and convert it to glucose, and the enzyme should be, you know what, let me just do this way. So we want glucose, and that should be glucose 6-phosphatase. Do you remember this enzyme? I think we talked about it earlier. Glucose 6-phosphatase. But the opposite is true too. We can take glucose and convert to glucose 6-phosphate in the liver, and that's known as glucokinase. Not hexo. Hexokinase is primarily in non-hepatic tissue. Hepatic tissue mostly have glucokinase. Okay.
This is in hepatic,
yeah? Yeah. In hepatocytes, yeah. Okay. So the one on the brown side is glycogen synthesis. This side is glycogen synthesis. Now you guys should know these enzymes. It's in your books. I'll write them small. So we have glycogen synthase and branching enzyme. Okay. On this side we have glycogen phosphorylase and debranching enzyme. So here you go. So let's focus on this now. Look. You get glucose in your blood from your diet, right? And this glucose is converted to glucose 6-phosphate within the cell. With the help of who? Glucokinase. Glucokinase. Next time, glucose can spontaneously be converted to glucose 1-phosphate. Spontaneously. It's spontaneous reaction by isomerase. And then glucose 1-phosphate, if you remember, do you remember that when I draw for you, glucose 6-phosphate can become glucose 1-phosphate, 6-phosphogluconate can become fructose 6-phosphate, and so forth. So going towards glycogen, glucose has to become what? Glucose 1-phosphate. As soon as you convert glucose 6-phosphate to glucose 1, it will become glycogen automatically. Now, the next enzyme is glucose 1-phosphate UDP transferase. Okay, and then you have the branching enzyme, and then you have glycogen synthase. Now, let's do this. I have to tell you something. Hopefully this makes sense to you. If it makes sense to you, it's good enough. You don't have to know the exact molecular details of it, right? This is what glucose look like. True? And it has phosphate at position 6. Okay? And let's add glucose in this form, because glycogen is nothing more than stacking glucose on top of one another. Okay? So you can make glucose attached 1-to-1, 1-to-1 position, which is not going to occur because we need the phosphate. Okay? So there's 2 ways you can make these bonds between carbon 1 and carbon 4, or carbon 1 and carbon 6. Okay, just remember that. Now if I let me twist it. If glucose is attached 1-to-4, it would look like this. 1-to-4. 1-to-4 gives you a linear molecule. True? Okay, so look, this is position 1, this is 4. So position 1 and position 4, carbon 1 and carbon 4. Glycogen synthase makes alpha 1,4 glycosidic bond, which means it attaches glucose 1 to the position 4. One glucose on position 1 and the second glucose position 4. Okay, so if I were to put them side by side, let's put it this way. Let me see if this will make more sense. So here you go, this is glucose, and this is glucose position 1, right? Position 1, and then there is another glucose, and this is the 4th position. So the O will differentiate. There's the O and there's the O, right? So you go clockwise from O, it will be the first carbon and so forth. So 4 here, 1 here. These 2. Now, if glucose also has the phosphate in position 6, let's bring that to position 1. You see that's why we have to convert to 1, because if you bring phosphate to position 1, that means it's become reactive. That part is now more reactive. Remember, we always talked about phosphate is what makes or gives energy to bond formation. So when you bring phosphate now, you can attach this glucose in this formation. So it actually removes, it will give off a phosphate and it will attach only by O. So this is 1,4 bond. 1,4 bond. Glycosidic, meaning it's between glucose molecules. Glycosidic. It's alpha. Alpha means it's opposite. It simply just means, okay, that it is attached in this way. The opposite ends. That's alpha 1,4. Alpha 1,4 glycosidic bond. Okay. Now, do you need to know alpha 1,4 glycosidic? I don't think so. As long as you understand glycogen synthase can only make alpha 1,4 glycosidic bonds. Therefore, it can attach glucose molecules end to end. So it'll become a straight chain of glucose molecules. Let's put it this way. So if you have this glucose molecule, you keep attaching them, they become a straight chain. Only a linear chain. If you want to store something, you have to stack them, right? You cannot store glucose as an individual glucose molecule. You have to stack them to make glycogen. Glycogen is a polysaccharide. Remember that? It's not a monosaccharide. It's not a disaccharide. It's a polysaccharide. Polysaccharide doesn't mean individual glucose molecules are just roaming around and swimming. It means they are all attached or bonded together. So glycogen synthase can make linear bonds. But now, in those cases when you have the phosphate at position 6, so you can add a glucose molecule at this position. So you can make a chain. You can make a chain. But this bond is alpha 1,6. So branching enzyme can make branches only by alpha 1,6 glycosidic bond. So they are the same thing in a sense. They both causes polymerization of glucose. That's true. You have a larger molecule of glucose, it makes a polymer. But branching enzyme makes branches only, whereas glycogen synthase makes linear molecule of glucose. So even you add here, the next one is in the linear fashion, the next molecule. So it's again, all blue bonds are made by who? Glycogen synthase. All red bonds are made by who? Branching enzyme. So branching enzyme can only do few. It's not a major one. So again, the first branch is made and then all added, and the elongation of that branch is by glycogen synthase. Yeah.
The difference between starch and starch because it's glucose molecule.
They're both glucose. Yeah. Glycogen is only made of glucose, not of fructose, not of galactose. In order to store fructose and galactose in the form of glycogen, you have to convert them to what? Glucose 1-phosphate. Glucose 1-phosphate. Okay? Alright. Now, it's just for you to make sense out of it. I don't think anybody will ever care for if it's glycosidic bonds or non-glycosidic 1,4, alpha 1,6. It's just for you to make sense. Why do we have two enzymes? Branching enzymes is not a major enzyme in storage of glycogen. It's not a major one. The major one is who? Glycogen synthase. Branching enzyme is needed, but it's not essential. Okay. Okay. So that's for this. Now, if you want to break, for example, now is the opposite side, green. If you want to break this glycogen, because we need glucose to restore blood glucose level, so you have to start breaking. Now, breaking linear, in a linear fashion, here, 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 here. Okay. That's done by glycogen phosphorylase. So glycogen phosphorylase breaks down alpha 1,4 bonds. Okay. But debranching enzyme can only break down which bonds? The branch enzyme, which is alpha 1,6.
The book says 1, 4, and 6.
Debranching? No.
Just take 1 section.
Okay. Okay. And then finally, glucose 6-phosphatase just removes the phosphate from glucose, and the glucose is no longer trapped in the cell. It goes where? Into blood. Glycogen synthesis should be promoted by insulin because it removes glucose from blood and deposits it where, or stores it where? In glycogen, liver or muscle. Now, breakdown of glycogen is promoted by who? Glucagon. And its job is to increase blood glucose level by breaking down glycogen. Okay, so this is, you should know this. Okay. These enzymes, I'm going to color it for you. The enzymes of glycogen synthesis and glycogen breakdown are found only in two places. Liver and what? Nope, that's not gluconeogenesis. Somebody said muscle? Muscle, yeah. Because you have only two sources of glycogen, two forms of glycogen, hepatic and muscle. So you'll find these enzymes only in two places, liver and muscle. Okay. Now, the only enzyme that's only found in the liver but not in muscle is this one. The one I'm going to color red. Okay.
It's found only in liver.
Only in liver. Because the function of liver is to what? Do what? Increase blood glucose level, right? The function of muscle glycogen is not to increase blood glucose level. It's to do what? To make ATP. So then in muscles, glucose 6-phosphate cannot be converted to glucose. It becomes straight to what? Fructose 1,6 bisphosphate. And this will become what eventually? Pyruvate. And then acetyl-CoA. And then ATP. That's what happens in muscles, because muscles don't have glucose 6-phosphatase. And they shouldn't, because their job is not to restore blood glucose level. Okay, you have a good picture in your book, so it's the same one. Look, all we care for is, do you understand how do you Do you understand the molecular biology of glycogen synthesis and glycogen breakdown? Yes, you do. 3 enzymes you have to memorize for glycogen synthesis, especially, and then 3 for what? For glycogen breakdown. That's all.
18.
Page 18. Good job. Thank you. Okay, now, are there conditions where these enzymes can be deficient? Yes. Okay, let's start with glycogen phosphorylase. Now you see this enzyme number 1. Let me just label it here, number 1. This one. It has 2 versions. 2 versions. Hepatophosphorylase and myophosphorylase. Write it down. 2 separate columns. So this glycogen phosphorylase has 2 types. One is muscle glycogen phosphorylase. The other one is hepatic or liver glycogen phosphorylase. I'll call it simple because for me it's easier. Hepatophosphorylase. Okay. There's one called hepatophosphorylase, and there's another one called myophosphorylase, MYO, and then phosphorylase. You could also call myophosphorylase muscle glycogen phosphorylase, and you can also call hepatophosphorylase as liver glycogen phosphorylase. So the enzyme glycogen phosphorylase have 2 different types. One is found in the muscle only, the other one's only found in the liver only. Is that okay? Good. Can I erase the cycle? Maybe I can put it to the side. Erase is a strong word. No good. Let's, there you go. So let's talk about those diseases that have to do with glycogen breakdown first. Okay. You'll be surprised how easy these diseases are. So hepatophosphorylase and then myophosphorylase. Okay, so hepatophosphorylase and myophosphorylase. Okay, there's the thing. They're both glycogen phosphorylase, true? Yeah, they're both glycogen phosphorylase. The enzyme known as glycogen phosphorylase have 2 types, hepatic type and muscle type, liver type, muscle type. Muscle type is also known as myophosphorylase, liver type is also known as hepatophosphorylase. Just multiple names, by now you should know these names. Okay. They both have one function, to break down glycogen. Break down glycogen to what? Glucose 1-phosphate. Their job is to do what? Produce glucose 1-phosphate. Produce glucose 1-phosphate. That's all. Sometime in this process, they will also produce branch points that they cannot break, and that have to be left for debranching enzymes. So now, what if you have a deficiency of these enzymes? I'll put a negative side beside them, and there is a condition that's found. Myophosphorylase is known as McCardle's disease. Okay, hepatophosphorylase is known as Her's disease. H for Her's, M for McCardle. Her's disease, hepatophosphorylase. McCardle, myophosphorylase. Would you have confusion? I don't think so. Not anymore. Easy to remember. Easy to remember. Okay. Now, when you have one of these deficiencies, let's start with hepatophosphorylase first. I just want to contrast them, okay? If you have hepatophosphorylase deficiency, is this a problem in glycogen breakdown or glycogen synthesis? Breakdown. And why would you want to break down glycogen? To restore blood glucose level. If you can't do that, what happens to blood glucose level? So you'll have hypoglycemia. Good? You will have hypoglycemia. Okay? Again, look, H, hypoglycemia. And hypoglycemia gives you headache, confusion, dizziness, and blah, blah, blah. Okay? Now, what do you expect? Oh, Her's, you see that it's H, Her's, sounds like female? Females are never affected. It's only in men. It's X-linked recessive disease. Her's is X-linked recessive. XR, X-linked recessive. McCardle is autosomally recessive. Yeah. Why am I silent now? I don't know. Okay, hypoglycemia. Now,
Autosomal recessive?
yeah, McCardle is autosomal recessive. Every other disease in this category is autosomal recessive except hers, which is X-linked. In myophosphorylis, what would you expect? Can you store glycogen? Of course you can. Can you break it? No. No. Yeah, you can say that. Myalgia. Myalgia. You see, the purpose of muscle glycogen is not to restore blood glucose levels, so you won't have hypoglycemia. So I'm going to write in big letters. No hypoglycemia. No hypoglycemia. Don't expect hypoglycemia. Euglycemia, normal blood glucose level, will be found in myophosphorylis deficiency or McCardle's disease. So the issue will be myalgia, muscle pain, Also, is this right, melon? Is that the right spelling? Melon color urine. I just like M's, that's why. It's my name, I guess, maybe. Red urine, hematuria, blood in urine. Not necessarily blood, but red urine. Just keep it that way. Okay. Okay. And then the feeling of lack of energy. What do you call that? Lethargy involves mental as well. Only muscle is malaise. Okay. So, feeling of lack of energy, lethargic, malaise, and then melancholy urine, myalgia. See, all M's. I mean, that's amazing. Wow.
So lethargy is incompatible with
No. No. We'll come to that. Yeah. You love lethargy. Lethargy can be lethal. No. Okay, so you see what you're going to focus on? Now, hers is always in children. Myophosphorylis in adults. I mean, yeah, myophosphorylis, McCardle.
Isn't it recessive?
It's recessive. It is found in children, but it's asymptomatic. In adults, it will be symptomatic. Okay? It'll be symptomatic. What's the primary? So let's talk about McCardle. What's the primary source of ATP for muscles? Fatty acid. Fatty acid. But when you work out, what happens? You need glucose. So McCardle is only symptomatic in men, not in children. In men. I don't know if a 14-year-old is a 15-year-old is men, not yet, but just think about it when they become aggressive in terms of exercise or start playing sports. So McCardle is only exercise induced. Exercise induced. So myalgia, melancholy urine, and fatigue will only come if you use too much energy. If your demand for energy increases but you cannot supply it. As long as you have, sources of ATP, your muscles will be okay. But when you exercise, what happens? You deplete muscle glycogen and you cannot break them down because of this. Okay, so fatty acid is not enough by that point. Otherwise, you can get most of your energy during the resting state. When you're resting, you're sitting, you're just relaxing. McCardle is asymptomatic. Does it make sense why? Only when you work out, when you do marathon running, when you play soccer, basketball, those high intensity sports, then the person will feel they won't be able to tolerate exercise. They're unable to tolerate exercise. That's McCardle. Okay. Now, so the myalgia you understand, because and malaise you understand, but melancholy urine, see, if muscle cells are not getting enough ATP and you're making it work, they will lyse. They'll go through necrosis. And the term is rhabdomyolysis. Rhabdomyolysis, some people call it myolysis, whatever you want to pronounce it. Rhabdo means bullet, rod shape cells, and muscle cells are rod, elongated cells. So rhabdomyolysis, and this will release something known as myoglobin, not hemoglobin. This is muscle hemoglobin, myoglobin. In myoglobin, remember, it carries oxygen, so it gets your cells red color or muscles red color. So if it's found in urine, because now you have too much, when it's lysed, it will come to plasma, and all of this plasma will be filtered where? In the kidney. So obviously you will have increased myoglobin in urine. And what do you call that? One more M. Myoglobinuria. So excessive amount of myoglobin in urine will give urine red color. So the person's exercise and then he feels fatigued and tired, lack of energy, muscle pain, and then he goes to urinate and his urine is bloody. He gets nervous. What did I do? Yeah. Okay. That's what it is. Okay. That's about it. Always in adults. Always exercise induced. When you're resting, no symptoms. Done. I think this was on my exam, Carl. I think I had this one for sure. I remember. Now, hepatophosphorylase deficiency. First of all, it occurs mostly in who? In men. It's a disease of boys. It's X-linked recessive. Girls are spared. Hers, meaning you get it from mom. Just think of it that way. Okay, but only boys get it. So you'll develop hypoglycemia. The problem here is you can store glycogen in the liver, but you cannot break it. Okay, same thing with muscles. You can store it, but you cannot break it. So muscles will enlarge, right? Myomegaly. So muscles will hypertrophy. They will enlarge. Floppy muscles. And as a matter of fact, then the glycogen can be converted to lipids in muscles too. But mostly what you... Just don't ignore this part. This part. Okay, don't ignore that part. This. So enlargement of muscle is not that important. Don't ignore these parts. Okay, let's move on to the opposite side. If you are storing glycogen in liver but you're not breaking, what happens to liver eventually? Hepatomegaly. Another H. Huh. Unbelievable. It's crazy. There are no coincidences, okay? I don't know, maybe it is. Okay. And the most you'll have is, okay, hypoglycemia, hepatomegaly. And then, another one, headache. Most blood glucose is used by what? Your head or brain. That's all. Okay. But there's the thing. When would this occur? Always in these children? No, only between meals. Even this is a mild form because what? In this case, you can make glucose from something else. You can make glucose from amino acids, right? You can make glucose from lipids. So there will be energy. So you might have hypoglycemia, but you might not.
But they have deficiency in lipids.
No, I never said that.
The kids.
No.
Deficiency to break down.
This is only deficiency of hepatophosphorylase. Hers disease is only hepatophosphorylase deficiency. Everything else is normal. So they can break down lipids to make glucose. They can break down fatty amino acids to make glucose. So they would not have hypoglycemia overall. It's only in extreme exercise in severe cases when they're deprived or they're starving. That's when you have problem. As long as they're eating properly, no problem. But the only issue is the liver can enlarge. And the enlargement of liver can cause problem. It can lead to jaundice and things like that. Okay? So, two major diseases. Okay, so we knocked out these two. Let's move on. You know, on this side, on the breakdown side, any enzyme deficiency will give you similar symptoms. Hypoglycemia, right? So we did this for the liver. Debranching enzyme. Debranching enzyme, that means you can produce less glucose 6-phosphate. But there will be some branch points, and that's known as dextrin. So there will be accumulation of dextrin. because you cannot convert it. Dextrin means about 10 carbon long sugars and things like that, or glucose. Okay. So you will have these dextrins. Okay, dextrins. You see, the reason you will have dextrin is this. Dextrin, dexa means 10, right? Or deca. Glycogen phosphorylase. How many carbon does a glucose have? 6, right? So this is one glucose molecule and it's branched to this chain. So that's 6 over here. And then 1, 2, 3, 4. Glycogen phosphorylase can only break up to 4. Beyond that, it cannot reach. It cannot reach to break this. So it's total about 4 plus 5 carbon sugar glucose molecules they cannot break. So you will have these dextrin having with these long chains. That's what it means, dextrin. Okay. So branch points. And the debranching enzyme disease is known as cutting disease. Debranching does what? Cuts branch points, right? So let's call it cutting disease. And the name of that is Cori. Cori, C for cutting. Cutting branch points. Okay. Cori's disease. And it should be deficiency of what? Debranching enzyme. That's Cori's disease. In Cori's disease, you might have, it's usually asymptomatic. Again, extreme activities might lead to symptoms. Extreme activities, okay? But it will be enlargement of liver like before. Liver will enlarge in all of these breakdown disease, all of them. Okay, so the difficulty comes, can you recognize the name? Glycogen phosphorylase deficiency, HERS or McArdle. Debranching enzyme deficiency, Cori's disease. Glucose 6-phosphatase deficiency, I mentioned it before. Von Gerke's disease. Good job. Thank you for being awake. Von Gerke's disease. Von Gerke's disease. Okay. So enlargement of liver. You should expect hypoglycemia. You should expect. Now remember, hypoglycemia in Cori's disease is mild compared to hepatophosphorylase deficiency. But even in hepatophosphorylase, it's moderate because you have amino acid and lipids that can be converted to glucose. Are we good? All of them should have hepatic enlargement. Von Gerke, guess what? Glucose 6-phosphate is required for gluconeogenesis. Even for amino acid to be converted to glucose, they have to go through this enzyme. So when you have von Gerke, it's severe. It is hypoglycemia all the time. Okay, especially between a child 3-4 months old, and then between feedings, they will be complaining of fatigue and being fussy and irritability and things like that, because this is true severe hypoglycemia. There's no amino acid that can compensate. There's no fatty acid that can compensate. Okay. Remember, what was the last enzyme of gluconeogenesis? Glucose 6-phosphatase. Yeah. So it's the same enzyme. It converts glucose 6-phosphate to glucose. So von Gerke, you cannot make glucose at all. There's a problem. You cannot make glucose in your body. The only thing you can do is get glucose from your diet. Okay, so as long as in all of these diseases, as long as the babies are fed properly, they should have no symptoms. But von Gerke is by far the most severe form of glycogen storage diseases. All these diseases are called what? Glycogen storage diseases. Because in all cases you store glycogen, too much glycogen. Either you can store too much, you cannot store it. Okay. Okay, let's move on to the other side.
I have a question.
Yeah.
Is there anything that you can give them to block the creation of glycogen?
Yeah. The best way not to store glucose is start eating small meals and frequently. So you don't have time of hypoglycemia and you don't have time of storage. Yeah. It's almost like day-to-day living. Don't live for like 100 years and store it for 20 years like, okay, I'll use it later. No. That's okay. Now on this side there's nothing is important. The only enzyme on this side that's important is branching enzyme. Branching enzyme, you can call it attaching. Attach. attachment or attaching enzyme. So the disease known as Anderson's disease. It's usually asymptomatic. The only thing would be the economic branch point. So you will have hepatomegaly. Okay, hepatomegaly, that's all. Nothing else. No hypoglycemia, nothing like that. Alright. We're done with these diseases. How much time did we spend? Okay, now I want to go to something. Let's talk about fructose metabolism, galactose metabolism, and glucose. Of course you know glucose. Glucose is first converted to what? Glucose 6-phosphate, right? By who? Hexokinase. Okay, and what's the specific form? Glucokinase. Very good. Similarly, galactose can be converted to what? Galactose 1-phosphate, not 6-1, but by who? Hexokinase, or specifically galactokinase. Okay. Similarly, fructose can be converted to, guess what? Fructose 1-phosphate. Only glucose is 6. Okay. And then hexokinase and fructokinase, specific one. Good. All 3 steps require what? ATP, correct? Because it's kinase. Okay. Glucose is further converted to reversible step fructose 6-phosphate, correct? And then it is further converted to what? Fructose 1,6-bisphosphate. And fructose 1,6-bisphosphate is converted to what? Glycerol aldehyde 3-phosphate. Okay. Okay. Now galactose can further be converted to, this was galactose, okay, Ga, will be converted to glucose 1-phosphate. So the, okay, well, I'm gonna make sure this G is glucose, okay, and this Ga is galactose, okay? Are we okay? Good. So galactose 1-phosphate is converted to glucose 1-phosphate, and the enzyme is known as galactose 1-phosphate uridyl transferase, or GUT. I don't know, there's the second GUT that we see. Okay, very important, so please memorize that. GUT stands for galactose 1-phosphate UDP transferase, or uridyl transferase. Okay, again, you have it in your books. Okay. Fructose 1-phosphate is then converted to glycerol aldehyde. The enzyme is known as aldolase. It's specifically aldolase B because we have A as well. And then glycerol aldehyde is converted to glycerol aldehyde 3-phosphate by trio kinase. Okay, so far they go in parallel. Everything is working, that's good. Now this will go and join what? It's the same thing as glycerol 3-phosphate, right? And from there it will be converted to what? 1,3-bisphosphoglycerate, and then eventually pyruvate, right? Let me see if I can make it smaller to make room. Oh, you know what? We'll make room later on. So once I finish this one. Now glucose 6-phosphate and glucose 1-phosphate. What's the difference? Phosphate at position 6 or 1. Do you remember that glucose 1-phosphate can be converted to what? G6P. It can also be converted to something else. Now, if you have too much ATP, glucose 1-phosphate is the first step in what? Glycogen synthesis. Good job. So it'll be converted to glycogen. If you have less ATP, it'll be converted to what? Glucose 6-phosphate, which is spontaneous reaction. Okay, so it depends what we do. What do you need? Energy or storage? Okay, so now galactose enters the glucose metabolism at glucose 6-phosphate position. Okay, whereas fructose enters in the glyceraldehyde 3 position. There you go, we have some room now. I just remember, I don't think we have this in the book. I remember Emily was asking me, yeah. So this is eventually pyruvate. It depends what you're going to do, ATP, storage, or whatever, right? Okay. Now, so we have discussed glucose metabolism already, not a problem. Let's go with fructose. In fructose metabolism, you could have fructokinase deficiency. Fructokinase deficiency. Do you think which one will be more common, fructokinase deficiency or hexokinase? Why should hexokinase be least deficient, least prevalent? Because everything depends on it. Yeah, so such thing don't exist. Okay, they have different isotypes of hexokinase anyways. So don't worry about that. Focus on fructokinase deficiency. If you have fructokinase deficiency, you will have fructose in urine, and it's a reducing sugar. Okay, reducing sugar. Oh, you know what? Please write down 2 things: reducing sugars and non-reducing sugars. Reducing sugars and non-reducing sugars. Yeah. All monosaccharides are reducing sugars. All monosaccharides. All monosaccharides. In this case, by all I mean what? The 3 that we have discussed. Galactose, fructose, glucose. They are reducing sugars. Okay? For example, if you use the enzyme known as glucose oxidase, fructose oxidase, or galactose oxidase, they will reduce these sugars and the color of the medium can change. So you can use it as a test. Okay? So reducing sugars. Disaccharide, lactose is reducing sugar. Maltose is reducing sugar. Sucrose is not. Sucrose is not. Sucrose is not reducing sugar, non-reducing sugar. Sucrose. Okay? Sucrose is non-reducing sugar. Reducing sugar means simply the rings opens up. It can open up. When you open up and you can attach them to like dyes and reaction, the color of the medium changes, you can test for them. At least you should know that sucrose is non-reducing. The other ones are reducing sugars. So for example, if somebody has sucrose problem and then you find there is non-reducing sugars in urine and blood, you will make only one conclusion. It's what? Sucrose problems or metabolism. Okay. Now, if you have fructokinase deficiency, it can lead to fructosuria. You know, a lot of fructose in urine because you're not able to metabolize it. Fructosuria, huh? Yeah, possibly, but not necessarily. Not necessarily. It's more for number one. The thing is, urine should be sterile. In diabetic UTIs are, for example, not common because it doesn't mean as long as bacteria doesn't travel from external sources, it won't cause. Yeah. Okay, so fructosuria. The second one is if you have aldolase deficiency. Aldolase deficiency. Now the problem would be you might not be able to tolerate fruit because the source of fructose is what? Fruit. So maybe you're not able to tolerate them. It will give you abdominal pain or lethargy and things like that. you know, so look for those things. Okay. Okay, galactosemia and galactosuria is more common. So galactokinase deficiency, you can have that enzyme, or you can have gut deficiency. In about 85% of all patients, it is gut deficiency. So galactose metabolism defect is most likely gut problems. Galactose 1-phosphate, UDP, transferase deficiency. Okay, that's the most common. So if I don't give you any indication in the question, and it is galactose problem, you have to think of what? Gut deficiency. It's more common. Okay, with galactokinase, galactose will build up. With galactose 1-phosphate uridyl transferase deficiency, you will have galactose 1-phosphate that will build up. Huge difference. Okay, so if you have gut deficiency, galactose 1-phosphate will build up in blood and in urine. If it's galactokinase deficiency, you will have deficiency of galactose 1-phosphate. So you can automatically identify them. Okay. Now, galactose metabolism, galactosemia, or galactose intolerance, or galactosuria, usually will lead to a byproduct of known as galactitol. Galactitol. When you have too much galactose 1-phosphate and galactose, it will be converted to galactitol.
Is there a disease?
Well, the disease known as galactosuria or galactosemia. Too much galactose in blood. Galactosemia. Yeah. Galactosemia have two causes. Galactosemia have two causes. Galactosemia. It simply means increased galactose where? In blood. Correct? So this cannot be lactose deficiency. That's a different thing. Lactose deficiency, you won't even have galactose in blood. You will have less galactose in blood. Now you have too much galactose in blood because what? You cannot metabolize. You can absorb it, you can bring it to blood, but you cannot use it. You cannot trap it in cells, you cannot use it. So it will increase in blood. So increase galactose in blood. Plasma galactose, okay? This is why the disease is called galactosemia. What are the causes? Galactokinase deficiency. That's one. Or galactose 1-phosphate uridyl transferase or gut deficiency. As I said, gut is more common. Galactose uridyl transferase deficiency is more common. So that's the more prevalent form. The most common cause of galactosemia is this one. I'll color it yellow for you. This is the most common cause of galactosemia. Now, in galactosemia, galactose will build up in blood. If you have too much galactose, it could be converted to what? Galactitol. 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 Okay, galactitol. It'll be converted to galactitol. Okay, so let me remove this because I think, yeah.
The galactose and the fructose, they will join the glucose to form pyruvate.
Yeah.
So if the diet is rich in galactose and fructose, but you don't eat like much glucose, it won't affect the cells.
No, glucose is easier to use. You can use galactose as a replacement, sure, you can use fructose as a replacement, but as I said, glucose is preferred. So it might not cause that many problems, but as long as you're eating. These deficiency it's what builds up. For example, in diabetes, so let's talk about two things here. Okay, I still wrote it down. Galactosemia. You know what? I'm gonna even make more room for myself. Let's move this guy. There you go. And you know that they join here, right? Okay. Now, let's talk about two diseases, diabetes and galactosemia. Okay. Galactosemia. That's one disease. And the other disease is diabetes. Diabetes mellitus or mellitus, but not insipidus. Okay. Now, in galactosemia, the deficiency is either galactokinase, right? That's the problem, or it is galactose-uridyl transferase deficiency. One of them. This is the most common one, of course. In diabetes, you have either insulin deficiency, no insulin, or you have insulin receptor deficiency. I'll put R, insulin receptor deficiency. At the end of the day, in diabetes, there is a buildup of what? Glucose. Where? It's plasma glucose that is elevated. So it's hyperglycemia, right? Is it okay if I write hyperglycemia? I mean, you're all educated, so you should have no problem. So diabetes leads to hyperglycemia. That's the problem. Because you cannot use glucose. You cannot remove glucose from blood. Now, in galactosemia, look at the name, galactosemia. It is galactosemia, hypergalactosemia. You have too much galactose where? In blood. Okay. So too much galactose in blood. Now, when you have buildup of galactose in blood in a tissue, this could be converted to galactitol. Galactitol. And glucose, again, I'm going to remove hyperglycemia now. I'll put glucose here. Too much glucose. Glucose is converted to sorbitol. Okay. When we have too much of them. The preferred pathway of glucose is to be used as glucose 6-phosphate and to be converted to pyruvate and so forth. If you can't do that, then obviously it will be converted to sorbitol. This is not a preferred pathway. This should not happen in normal people. Galactitol is not normal. It shouldn't happen in normal people. In small concentration, your body can deal with it. In high concentration, your body cannot deal with it. Yeah.
Does that carry sorbitol, or is it just total toxic?
In high concentration, it could be toxic. But in small concentration, it's converted back to glucose and it's used. Okay. Now, galactitol is toxic, especially to neurons. Okay, neurons, lens of your eye. Sorbitol is that toxic to who? Retina, nerves. Now, when I say neurons, it's peripheral neurons more than central. Okay. So nerves, I'll use the term nerves. And, of course, glomerulus, right? Glomerulus. Muhammad, you were saying something? Yeah. Okay, or blood vessels. So now, in galactosemia, you should say there is a chance of cataract because of lens problems. Cataract formation. And then there is a chance of failure to thrive, meaning the baby doesn't build properly or does not grow properly. Okay, and there's a chance of hypotonia, muscle tone is affected, and there's a chance something over there, but this is an adult disease, right? Diabetes is an adult disease. Galactosemia is disease of children. It is deficiency of enzyme. Diabetes, there is no enzyme deficiency. It is what? Insulin problems. Okay. And then, of course, mental retardation is possible in mental problems because galactose is needed for myelin sheath formation. Okay. That's what you need to look for. Other than that, there's nothing. But of course, diabetes is much larger disease, so we have to focus on the effect of diabetes overall. It causes retinopathy. We discussed it, I think, the other day. Did we discuss this or no? So I don't want to go too much into details because they will be overwhelming you. as long as too much glucose is not good. Okay, so look, you have to know fructose, urea, galactosemia, okay, and diabetes. Let me compare them now differently because I have room. And I'll spend more time on diabetes. What, do I need to take a break or no? How long has it been? Let's take a break.
Like the enzymes.
Oh, how was the practice exam yesterday? I thought it was excellent. Sure, we'll finish. I think we'll finish early. Okay. Can somebody email me that one? Is it in PDF or is it in? Because I want to change it. I can copy and paste, right? I should be okay. I'm okay, right? Stop. Okay. All right. Now let's start with one crazy glucose metabolism pathway known as pentose phosphate shunt. Pentose phosphate shunt pathway. In this pathway, glucose 6 phosphate is converted to 6-phosphogluconate, and 6-phosphogluconate is converted to ribulose 5-phosphate. Okay.
Ribose?
Yeah. Initially ribulose and then becomes ribose. So let's just call it ribose. That's fine. Okay. Here is an enzyme of extreme importance to us, especially to Ahmed. Yeah, for you. I'll tell you why. See if you can make sense out of it. No, nothing to do with that. So, it's just because when he was a child, he traveled from Afghanistan all the way to Egypt on feet, and he's supposed to be Afghan national, but now he's Egyptian, so it has to do with that. I told him stories, and I love that kid. He makes me laugh, so that's why. So the enzyme here is glucose 6-phosphate dehydrogenase. Okay. The second enzyme is 6-phosphogluconate dehydrogenase. Please memorize these, especially the first one, Ahmed. Yeah, especially. The reason I'm saying is this deficiency is common in Egyptian. Very, very common in Mediterranean people, which includes, let me come to the disease, I'll tell you. Okay. Now, okay, so here, if it's dehydrogenase, what do dehydrogenase use? Always NADH or NADPH or FADH, one of those. But in this case, it's NADP is converted to what? NADPH in both cases. NADPH. So what is it generating, this reaction? This reaction is generating something big thing, which is very important for us. NADPH. This is what we care for. This reaction produces what? NADPH. This is a huge source of NADPH. These two enzymes are located where? In the cytoplasm. Memorize that. They're not mitochondrial enzymes, they're cytoplasmic enzymes. Okay? So that's one thing. Okay, ribose 5-phosphate. You can use ribose 5-phosphate because it looks like this. And if you see the picture, you'll be like, oh, I get it. Yeah. This is used for nucleotide synthesis, DNA and RNA. Of course, RNA first, because it's ribose, right? Ribose have to be converted to deoxyribose. What's the enzyme that converts ribose to deoxyribose? ribonucleotide reductase. Memorize that. Cannot ignore it. Which we discussed in DNA and RNA. So anyhow, question?
No.
Okay. So we can use ribose 5-phosphate to make the sugar, donate the sugar to DNA and RNA. Okay, and it can further be converted back to glucose. And here we need an enzyme known as transketolase. Transketolase. Ketolase. Okay. Transketolase. Okay. Now, what's the big deal? NADPH is anabolic molecule. It is needed by reaction that requires a lot of energy. NADPH is a huge source of ATP, much bigger than NADH. It's a high energy compound. So anabolic reactions requires these guys. Anabolic reactions. Now let's say, what are these anabolic reactions? When you make bile salts,
Cholesterol.
very good. Cholesterol.
Steroids.
Okay, cholesterol and steroid are the same thing, right? Good. Okay, fatty acid synthesis.
Vitamin D.
Vitamin D, steroid, cholesterol, same thing.
Same thing.
Yeah. Okay. Soon, I think Friday you will learn about lipids, that the source of vitamin D, bile salts, and all of these and steroid hormones, especially steroid hormones and and vitamin D, is cholesterol. Cholesterol is the source of them. Okay. So all these reactions, conjugation of bilirubin and blah blah, all these reactions requires a lot of energy. Even DNA synthesis requires NADPH. Protection or production?
Production.
Say the other one.
Protection.
Protection. There you go. Protection. Okay. All right. So now, guess what? There's other enzymes. So this whole cycle you want to memorize for only one thing, and that is for NADPH. The purpose of this shunt or pathway is only to understand what is the role of NADPH. Okay. NADPH reduces glutathione. Okay, let me write it down. Can I erase this now? Okay. I'm gonna do it. Okay. Glutathione is a molecule that is supposed to detoxify. This guy detoxifies. Detoxify free radicals.
Especially important for acetaminophen.
Corn syrup?
No, acetaminophen.
Acetaminophen, yeah. In all of them. Yeah. Glutathione will cause detoxification of free radicals, okay? Now, so that's one thing. Free radical exists as a reactive oxygen species. For example, this is superoxide. Superoxide. That's superoxygen. That's one radical. Hydrogen peroxide. Another one. This is another one. Hydroxide ion. These are free radical. And then you have perchlorate. Okay. These are free radicals. These guys are free radical. Well, the dot literally means free radical. Now, free radicals are toxic to human body. But they are, guess what? Normal products of what? Metabolism. But you know, your metabolism is not so perfect. So guess what? In the process, you produce these free radicals, and then glutathione is supposed to detoxify them. Good?
Only glutathione?
Okay. Glutathione. There's other ones too, but it's the major one. Glutathione and peroxisomes is the major one. Okay, so glutathione will detoxify them and then neutralize them. For example, convert. We have catalysts that will convert hydrogen peroxide into what? To water and oxygen. Yeah. Okay, so there's other ones too, but glutathione is important for them. Okay. Now, glutathione reduction requires, or activation requires, NADPH. NADPH. NADPH is needed for glutathione production. Okay, glutathione production. You need NADPH. If you don't have NADPH, what happens to glutathione? It's mostly oxidized form, and it's not able to neutralize free radicals. So in essence, the only purpose of NADPH right now we can think about is detoxification. Detoxification. Good? So think for that. The other source, the other purpose of NADPH is to make cholesterol, bile salts, fatty acid, or fatty acids, you know. So two things, NADPH. Detoxification. So NADPH has a role in detoxification. It's a detoxification and anabolic reaction. Not catabolic. Anabolic means building larger molecule. Those that require high energy. So that's the two purposes of NADPH. Yeah.
I have a question.
Mm-hmm.
Is NADPH, does it reduce glutathione or does it, is it used for the production of glutathione?
Which is the same thing.
Okay.
So glutathione in the oxidized form is inactive. It cannot help in detoxification. So at the end of the day, it's the same thing. It just gives the hydrogen ion to glutathione. Okay, so the hydrogen ion is donated. It's carried by these guys. So again, they're nothing more than a carrier, right? So if you worry about the molecular biology of it, it simply gives the hydrogen ion, not a proton. Hydrogen ion is hydride ion, like negative hydrogen ion. It gives it to what? To glutathione. And glutathione can now actually cause neutralization of free radical. If you don't give the hydrogen ion, then glutathione can't do anything. It's inactive in a sense. Okay? That's the only difference. Okay? Now you can have inactive glutathione all you want. It's not functional for this purpose. Okay? But glutathione is made of glycine and cysteine and glutamine if you care for it that way. But that's irrelevant. Irrelevant. You can have a knife, you know, that is covered with a case. You can cut anything with it. So you have to remove the case. Okay. Now, so the enzyme that produces NADPH, one of them was glucose 6-phosphate dehydrogenase. Correct? That's one enzyme that produces NADPH. Correct? What does it produce it from? Glucose 6-phosphate, and it gives you 6-phosphogluconate. Correct? There's one more enzyme, isocitrate dehydrogenase. And isocitrate dehydrogenase, what do they do? It takes isocitrate and converts to what? Alpha ketoglutarate. Very good. Okay. Okay. Another one is glutamate dehydrogenase. Glutamate dehydrogenase enzyme. Glutamate dehydrogenase takes alpha ketoglutarate and converts to what? Glutamate. Okay. So these are the enzymes. Takes alpha ketoglutarate, converts to what? Glutamate. Now, all of them produce what? NADPH. But do you remember that when you talk about isocitrate dehydrogenase, it was able to, in the Krebs cycle, it only produced what? NADH. There's a form of it, the second version of it, who can produce NADPH. Okay? It's the same enzyme but has different version of it. These are the three enzymes that I know of that can produce NADPH. Okay. What is the location of glucose 6-phosphatidehydrogenase? This enzyme. Where would you find this enzyme? Only cytoplasm. Thank you. Only cytoplasm. Very good. How about isocitrate dehydrogenase? Different color. Only mitochondria. How about glutamate dehydrogenase? Mitochondria. The only one that's cytoplasmic is what? Glucose 6-phosphatidehydrogenase. Now,
Wait, N6PG.
yeah,
Glucose 6-phosphate and 6-phosphogluconate.
6-phosphogluconate dehydrogenase.
They're both in the cytoplasm.
Yeah. Okay. Now there's the issue. The issue here is there's no such thing as isocitrate dehydrogenase deficiency, glutamate dehydrogenase deficiency. They are severe. They don't exist. If they are, it's small amount and people usually don't survive them. But there is such thing as known as glucose 6-phosphate dehydrogenase. Okay? It's also known as glucose 6-phosphate dehydrogenase deficiency, right? Okay. Now, if you have deficiency of this enzyme, what is it? What's the problem? Number one, this enzyme gene is located on the X chromosome, so it's X-linked. recessive. It's inherited in XX recessive fashion. So who's affected by this? Boys. Okay. So that's first thing. Where is this deficiency more common? In Mediterranean areas, which includes Italy, Portugal, Spain, Egypt, Algeria, North Africa, and South Europe. Okay, so those areas. It's common in those areas. It's prevalent in those areas. Okay. Now, if you have glucose 6-phosphate dehydrogenase deficiency, so is that a big deal? If it is, please tell me why. If it's not, let's move on.
Yeah.
Why is it a big deal?
Anemia.
Why anemia? Why hemolytic anemia? Why rupture of RBCs?
Free radicals. Increase free radicals. No mitochondria.
There you go. Now we're talking. Well, he just said that these two are found in the mitochondria. RBCs don't have mitochondria. So if you have glucose 6-phosphate dehydrogenase deficiency, it doesn't mean it's only deficient in the RBCs. It's deficient in all cells of your body. But only RBCs are affected. Why? Because they're solely dependent on glucose 6-phosphate dehydrogenase for generation of NADPH, since they don't have mitochondria. RBCs, one more time, just like we talk about pyruvate kinase, RBCs don't have mitochondria. Therefore, they cannot generate NADPH from mitochondria. They don't have it. They only have one source of NADPH, and that is glucose 6-phosphate dehydrogenase. Make sense? So therefore, by this deficiency, only RBCs are affected. Any cell that has mitochondria is not affected by it, because those cells will get NADPH from other sources. They will get it from isocytosine dehydrogenase and glutamate dehydrogenase. Not a problem. But RBCs don't have mitochondria. They don't have these enzymes. They only have glucose 6-phosphate dehydrogenase. If that enzyme is deficient, there's nothing for them. So in RBCs, there is lack of NADPH. Other cells will not have this problem. Only RBCs would. Now guess what? So if RBCs are unable to produce NADPH, who cares? The only thing is they cannot prevent free radical damage. So now let's move on. I hope you understood this part, okay? I'm going to erase it. This is a big disease. It always comes on the exam. Always, always. Always look for boys being affected. Always from Mediterranean area. At least on the exam. I'm not saying it doesn't exist in me or my ethnic group or other ethnic groups. It is endemic, or it's more common, it's more prevalent in Mediterranean area. Just like diabetes is more common in Hispanic here. Just like hypertension is more common in African American here. Just like sickle cell anemia is common in African American, and just like cystic fibrosis is common in European, and just like Crohn's disease is more common in European and Central Asians. So there are these factors. So that's why they're going to use the ethnicity and gender always. Okay.
What is the disease called?
Glucose 6-phosphate dehydrogenase deficiency. Yeah. But you can call it anemia. Hemolytic anemia. But this hemolytic anemia is caused by what? It's not caused by pyruvate kinase. It's caused by glucose 6-phosphate dehydrogenase. So that's what the disease is called. Glucose 6-phosphate dehydrogenase deficiency. Anemia. Okay. Now, let's say how it develops anemia. Look, once we have RBCs, they do cause metabolism of glucose to generate ATP. But also other cells are producing free radicals, and the free radicals are flowing freely in plasma. RBC don't have any NADPH to protect themselves from these free radicals. So now free radical can cause damage to RBCs. So RBCs are more prone to free radical damage. That's all there is. They're more prone, so then they can cause damage to them. Free radical damage, meaning that these free radicals cause, and what do they do, free radicals? What do we call that term? Please, there's one term for lipid. Lipid per oxidation. This is what free radical do, and it's a chain reaction. It keeps going non-stop. Lipid peroxidation. Please memorize that. Free radical causes lipid peroxidation. Now, the plasma membrane in this case, let me do this. The plasma membrane of RBCs is made of what? Lipid bilayer. So it causes damage to this lipid bilayer. And when it's damaged, some molecules will get exposed on the surface of RBCs, and now complements will come and attach to them. Complements. And the complements will, they're like knives. They will punch a hole in the RBC and lyse them. So it's hemolytic anemia. Now, glucose 6-phosphate dehydrogenase is hemolytic anemia. Now, hemolysis is the term. If I'm going too fast, slow me down, because I want to go over that exam. This will be the last thing for today. We're done with glucose metabolism after this. So the term hemolysis simply means bursting, rupture of RBCs. Rupture of RBCs. Two places this can occur. One is within vessels, which is known as intravascular. The second one is in spleen, primarily spleen in adults, some in liver, some in lymph node, some in bone marrow, and these are known as reticuloendothelial organs or tissue. But I'll keep it to spleen for now because that's the primary one. And spleen, it's done by the help of macrophages. Macrophage. In blood vessel, it's done by who? Complements. Complements, specifically, it's known as membrane attack complex, or MAC. Return of the Mac. Do you remember that song? It's an old song. Yeah. Okay. So macrophages and membrane attack complex. At the end of the day, when you burst RBC, when it dies and it ruptures, but now, is there anybody who doesn't know what RBC is for? All of you guys know what's the function of RBC. What is RBC for? To carry oxygen. What carries oxygen? Hemoglobin. So RBC is full of what? Hemoglobin, hemoglobin, hemoglobin. Obviously, if it ruptures, what happens to hemoglobin in blood? It swells and will increase. Even if it's done in spleen. Spleen is also vascular, so all the hemoglobin will accumulate where? In blood, regardless. Now, hemoglobin has two parts to it. Hemo and then globin. Okay, after hemoglobin is released in blood, since it's toxic, then your body and your liver and your spleen, the macrophages will take this and separate them. It will separate them into, the hemoglobin will be separated into globin and into heme. Globin is a protein and then it's converted to amino acid. But heme is a toxic substance. It's water insoluble. Further, in the macrophages and in blood, it's opened up. The ring of heme looks like this. It looks something like this. In the center of it, it has iron. And this ring is opened. Look, it's heme right now. If you open it, it will look like this. Okay? When you open the ring, it's still insoluble. Now, but the open ring is known as bilirubin. Bilirubin. Billy and Reuben got together. And then they made something like this. Okay? So this, now, so that's bilirubin. Bilirubin, if it deposits in your skins, it will give you yellow color, jaundice. So deposition of bilirubin in your skin will, so it will give you jaundice. Jaundice is caused by because of what? Too much bilirubin being deposited in your skin, in conjunctiva of your eyes, and so forth. Okay, so jaundice. Bilirubin should be conjugated by the liver. Conjugation means you add a sugar to it. Glucose, UDP glucose. When you add UDP glucose to bilirubin, it becomes conjugated and it's more water soluble now. So we have two types of bilirubin. Conjugated and unconjugated. Okay, so bilirubin is two types: unconjugated and conjugated. Unconjugated is very toxic. It's insoluble. Conjugated is still mildly toxic, but it's water soluble, so you can pee it out. You can get rid of it. You can get rid of it in stool. Now, if bilirubin gives stool brown dark color, so when you have too much bilirubin and it's not being conjugated, so some of it will go where? In urine. So it'll make urine dark color. And stool will stay light color. Light stool. So that's typical of all hemolytic anemias. Doesn't matter what causes the hemolysis, the result is the same. So in glucose 6-phosphatidehydrogenase deficiency, we have deficiency of an enzyme known as glucose 6-phosphatidehydrogenase. It's X-linked recessive. Who's affected? Boys. So they will carry this mutation for the rest of their life. Now, the only time you'll have symptoms if there's hemolysis. If there's no hemolysis, no symptoms. It's silent. No symptoms. When would you have more hemolysis? When you're under stress, infection, acidity. When plasma acidity increases, so the risk factor, let's talk about triggers now. Look, glucose 6-phosphatidehydrogenase carriers are not always symptomatic. So it is episodic disease, and there are triggers for it. And who are the triggers? Acidity, let's say low pH. Now this can usually happen when you sleep and your mouth is closed and carbon dioxide keep accumulating. Plasma acidity. Infection. Infection. So infection will cause it. Fava bean, yeah? Because it produces a lot more free radical. That's the unique thing about them.
NSAIDs?
Huh?
NSAIDs?
No. Let me tell you this. Sulfa, methoxazole, and trimethoprim. This drug will cause it. Which part of it? This is the sulfa drugs. Sulfa drugs. Sulfa drugs. Now, these are all triggers for them. If you don't have these, then you're normal. No problem. But if you... Now, what if somebody has infection? Bacterial infection. That's one risk factor. And on top of that, you use sulfa drugs. That's two risk factors immediately. Usually the question tells you that. Oh, somebody had an infection, blah, 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 and you give them sulfamethoxazole and this, that, and then he developed a pale skin color, pale stool, dark urine, and jaundice. What do you think? And then you have to figure out what part of the cycle they're asking about. Okay? And it's always boys. So, again, there are triggers for that. Unless your body is under stress, this will only cause symptoms when you have too much free radical production. Too much free radical, not normal free radicals. Look, if there's normal concentration of free radical, that means mitochondrial enzyme will neutralize them. No problem. But when you have so much free radicals that your body cannot accommodate them, of course, RBCs are at risk. Otherwise, they're not. So it's always episodic disease. Always episodic disease. Okay, so it depends. It's 100% guaranteed on the exam. Depends what they ask. They ask about the biochemistry of it, the anemia of it, you know, the drug, how you prevent it, and so forth. Okay, so look for those things. But at the end of the day, it's nothing more than hemolytic anemia in boys. Hemolytic anemia in boys. Episodic hemolytic anemia in boys. Triggered by what? Infection or medication use or fava beans. Now, fava beans is more common again in North Africa, so it's a major dish. Or yeah, is it appropriate to call it dish, or is it just like... Okay, so that's for this. We are done with glucose metabolism. What time is it? 10:37. The last thing I want to do is lactose intolerance, which we can do in the GI. I'll leave it for GI. Okay. Huh? Yeah.
I had a childhood friend who didn't eat solid beans, and I